like to introduce the final speaker of this session, Paris Bettencourt.
Ladies and gentlemen, good afternoon. I'm Claire from France, and these are my teammate Julian from the United States and Aisha from Indonesia. So we are happy to present our project, Beware, How Safe is Safe Enough? You might wonder how 9 university students got interested by biosafety in the first place. Well, what came out from our first brainstorming session was that most of us wanted to make the Earth a better place. We came up with this idea of a DDT degrading bacteria, the first good step to Earth depollution. Other step might be depolluting water from plastic and heavy metal. However, this project had its downfall, as we might easily lose control. What if the bacteria proliferate so excessively that they outcompete the water bacteria? What if there is hostile gene transfer and the whole ecosystem ends up contaminated? Then what? Well, then we don't know. So you can see there are many concerns arising from the release of genetically modified bacteria in the environment, and this problem concerns the general public as much as the scientific community. And this is why we decided to put human practice first. As you can see from this graph, biosafety concerns has actually been growing in the iGEM community over the years, and some teams tried and addressed these concerns. But how safe is safe enough? We tried and addressed this question through research, drawing guidelines for better practice in synthetic biology. So our approach was the following. We listened first. So we got input from various sources. We interviewed experts. We organized a workshop. We organized the debates. We screened the literature to know the state of the art. And we screened previous iGEM Teams wiki looking for safety modules. So the workshop, so we invited 60 high school students to our lab and we trained them in synthetic biology. Then it was their turn. They had to pretend they were an iGEM team and come up with a project. And then they had a class discussion on benefits and risks. And we were surprised by the high level of the discussion. Then we organized a workshop that over 100 people attended. In the first part of the workshop, 10 university students, but none having a training in synthetic biology, had a pro-con debate on the following motion. This house would allow environmental release of genetically modified bacteria in the following field: medicine, bioremediation, agriculture, and biofuel. And then the debate was open to the floor, which enabled us to have a broad range of input as the public was so diverse. So then we screened previous iGEM Teams wiki, so since 2006, looking for safety modules, and we identified 32 teams that actually had safety modules. So these modules fall in 3 categories: kill switch, physical containment, and aggregation modules. So then we synthesized and analyzed all of this information in a 60-page report that you can find on a crowdsourcing platform. So we invite all of you to come, read the report, and then modify it to make it better. So all the conclusions can be found on the report, and I will now run through the main one. So first of all, hostile gene transfer is one of the main concerns in terms of safety for the public and for the scientific community. Therefore, it should not be ignored. Secondly, absolute safety is not possible, but people are not asking for zero risk. They are willing to accept different level of risk according to the different level of benefit they foresee. Therefore, each project should be assessed on a case-by-case basis and addressed with the public from the start. So this brings me to my third point. If we want the public to be able to weigh risks, safety modules need to be accessible, characterized, and quantified. And last point, we wish that in the future, every team that creates a bacteria that needs to be released in the wild can embed the safety module in the projects from the start, and that this will be made very easy by the existence of standard plasmid containing those safety modules. So in an attempt to address some of these conclusions, we created a safety page where we list all of the existing safety modules, and we created a safety assessment protocol that Aisha will detail later, enable for people to be able to characterize and quantify these modules. So now we are asking for major efforts in the iGEM community. We're asking teams to come to our page and use our protocol to quantify these existing modules, first of all, and second of all, to create new modules with characterized and quantified information. So now that we have a very strong human practice background, we are ready to hit the bench.
Thank you, Claire. So when we went to the bench and we thought about our containment modules, we wanted something that would be robust. So our containment needs to be redundant, meaning it's more than one mutation away from failure. It needs to be modular, which means it can fit with your favorite iGEM project. It needs to be efficient. It needs to do what it's supposed to do. It needs to be quantified so that we can predict risk. And of course, it needs to be harmless to the environment. So that's why we came up with Beware, a library of modular devices that can be plugged into your favorite iGEM project. So I'll overview these now. We first have physical containment. Then your project needs a time frame on which it can work, for which we need a delay. This is followed by a kill switch based on DNA degradation and population level suicide. Finally, we have semantic containment, where your genetic information is encrypted in a language that can't be read by wild-type populations. So physical containment is our first barrier between our genetically modified bacteria and the environment. So we wanted something that would allow us to control the environment of our bacteria, the population size, and something that could be easily manageable by the user. And so we modified a protocol using alginate capsules to encapsulate our bacteria. And so we have simple alginate capsules, which you can see on the top. And we also developed a way to chemically treat these capsules, creating covalent crosslinks, which dramatically reduce the escape rate of our bacteria on the bottom. And so first we asked, could our bacteria survive in these capsules and perform normal metabolic function? So we tested the activity of beta-galactosidase in the presence of xGAL. And we see the normal expression in our reduced and our induced states. And here we also have a nice visual indication of our escape in our untreated beads and our treated beads. And so we see the supernatant is blue in our untreated cells, so we hypothesized, in our untreated beads, so we hypothesized that our cells were escaping. So we wanted to quantify this further. So we simulated a harsh environment in which we placed our untreated and treated beads, and we were able to have up to over 99.9% containment of our bacteria. But of course, if we're working with a very large population, we need more containment. So this brings me to our next module, our kill switch. So for our kill switch, we decided to work with wild-type colicins. So colicins are toxic proteins that are actively imported into E. coli. So that means that they are work on a population level and they're target specific. So we chose colicin E2. It's a DNase, which means it degrades bacteria. And in these wild-type populations, they also produce an immunity protein which tightly binds the DNase, protecting the producer cell from this DNase activity. So we thought we could take advantage of this system and place our toxin on the chromosome under the constitutive promoter. We've placed our immunity protein on our Beware plasmid. We also have in green your plug-in project. In addition, we have our restriction enzyme under the repression of LAC-I, and our Beware plasmid contains specific restriction sites for our restriction enzyme. So let me just walk you through this. In the presence of arabinose, LAC-I is produced, and our restriction enzyme is repressed. Our immunity protein is produced, and your cell can perform some function. Arabinose diffuses from our capsules. We have the decrease in our LAC-I activity. Our restriction enzyme is de-repressed. Our restriction enzyme can now cut our Beware plasmid, which is then degraded. We no longer have production of our immunity protein, and our DNase is now free to act on the cell, degrading the genetic contents of our cell and killing the cells. And I remind you that colicins act on a population level. So that means that cells that mutate their toxin are not able to escape this kill switch. So we have population level suicide, and our free colicins can degrade free DNA left in the environment. So this system answers the problem of mutants that escape by mutating their toxin, as well as horizontal gene transfer. And so I'll go through the experiments to test our system. So first, we characterized our delay. So I remind you, our delay is based on the exponential diffusion of arabinose and repression cascades. So here we have a GFP reporter in place of our restriction enzyme. So first we incubate our cells in arabinose. We then wash them and resuspend in either arabinose, in plain LB, or in IPTG. And so we see if we keep them in arabinose, we don't see any expression of our GFP reporter. However, if we allow the arabinose to diffuse, or by IPTG, we see expression of our GFP. And the difference between the IPTG, the delay, and the simply allowing the arabinose to diffuse is our program delay due to the degradation of black eye. Now, if you combine all these delays together, you get the total delay. We also characterized a new promoter, P-rhamnose. So this is orthogonal to P-TED, P-LAC, and P-BAD. So this is modular, it can be plugged into your favorite project. And so we placed our P reporter under our P-rhamnose, and what we see is that in the absence of rhamnose, really a tightly controlled, no expression of our reporter. And here we have our induction as a function of our concentration of rhamnose. So we hope this new promoter will be useful for you. So now for our restriction enzyme system, we chose the yeast meganuclease ICE1. So on one plasmid we have our meganuclease produced under control of P-LAC, and we also have chloramphenicol resistance. On a separate plasmid we have ampicillin resistance and our recognition restriction sites. So when we incubate these cells in chloramphenicol and we then plate on chloramphenicol and ampicillin plates, if we don't induce our system, we see about 20% of the cells retain our ampicillin plasmid. If we induce our system, we see a significant decrease in the number of cells that retain the ampicillin plasmid, indicating the restriction enzyme activity. And of course, in our first steps for our synthetic colicin system, we need to provide immunity to our cells. So if you plate wild-type cells, and you spot with colycinogenic cells, you see a zone of clearance around the colycinogenic cells where they have killed the wild-type cells. Now, if we transform our wild-type cells with our immunity plasmid, we plate our immune cells, we spot with colicin, we no longer see our zone of clearance, indicating that they're immune to the colicin activity. So we wanted to quantify this further. So we mixed our immune cells with colicin extract, which you can see in green. They grow normally like our controls, and this is in sharp contrast with sensitive cells when mixed with colicin in red, which simply cannot survive. So these colicins have a lot of really interesting and useful features, one of which Aisha will go into now.
Okay. Thank you, Julian. So as Julian already said, the colicin toxin is actively imported into E. coli. So we came up with this idea: can we hijack the properties, the import properties of colicin for synthetic biology? So if we can swap the colicin toxin domain with any protein of your interest, we can create a synthetic import domain which can have many interesting features. We can have a new cell-to-cell communication system, or we can even make our beware containment system more robust by importing restriction enzyme. So we tested this idea by doing a complementation assay. So we have producer cells which has synthetic import domain and Lexi alpha, and it's controlled under IPTG. And we also have our receptor cells which contain Lexi omega. And if this synthetic import domain, if this synthetic import domain is importing Lexi alpha into the receptor cells, Lexi alpha and Lexi omega will form an active enzyme, and if we put Xgal into the cells, they will turn blue. So we had a promising preliminary result when we mixed the producer and receptor cells and we use with IPTG. Here we can see clearly a high blue expression, and it's not seen in our control. So this is good, and we hope we can go forward from it. But now, let's go back to our containment system. This is the third level of containment system, which is a semantic containment. So in case of horizontal gene transfer, we don't want our genetic engineered gene to give advantage to natural strain. So we did, as a proof of principle, we modified a plasmid which gives kanamycin resistance by replacing serine codon with TAG, which is a stop codon. In our engineered cell, we have sub-T, which is tRNA under pressure, which can recognize the site and give kanamycin resistance. But if this plasmid is transferred into the natural strain without sub-T, the cells will read TAG as stop codon, so it won't give kanamycin resistance. And then this is our experimental data. We grow cells in different concentration of kanamycin, and as we can see in the positive control, the resistant strain with normal kanamycin resistant gene, they grow in any concentration of kanamycin. But our sensitive strain without kanamycin resistance, they won't grow after passing certain threshold of kanamycin. And now we mutate one amber codon, and we can see the cells now have sensitivity to kanamycin. But we can still see the difference with the sensitive strain, so we were not satisfied with this. We mutate two amber codon, and now our cell have the sensitivity almost the same, or the same with the sensitive strain. And if we put the plasmid into the cells containing sub-D, we can see now the cell restore the kanamycin resistant. So it means our system worked. And the idea is we need to put semantic containment into any genetically engineered gene before we release them into the environment. So now we already have different containment modules, and we want to know how good they can protect our cells or our system. So we try to do a safety assessment. We try to do adaptation of safety assessment method that already exists in safety engineering. It has been widely used to minimize the risk, and this is our step-to-step protocol. First, we identify what are the hazards, which are the out-competition of genetically engineered bacteria and also horizontal gene transfer. And from this, we did a safety risk assessment, and then we designed some controls to control the risk. And lastly, we check the control, and we try to make a quantification of these controls. So what we did is we did a fault tree analysis to see the relationship with one module and the other. And this is important to see what are the key elements that can make our system fail. So for example here, our genetically engineered bacteria outcompete natural strain can make our system fail, or horizontal gene transfer happen and semantic containment fails. And from this, we can go on and on to have more details until we have a list of basic events, and then we predict the probability of each basic event to happen. And from with AND and OR gates, we can predict the overall failure rate. And from our calculation, we come to the number in order of magnitude 10 to the minus 11 accidents per cells per generation. And the important thing here is the failure rate is given in our unit of per generation, which means that we need to take into account how many generations we want our system to work, and also what are the population size that we want. So we realize that this is still a rough estimation, so we invite you to come and see our protocol in the biosafety page in the registry, and use this protocol to analyze your system and improve it towards better understanding of safety in synthetic biology. Okay, so now I would like to zoom out and outline our achievement. In the beginning of the summer, we received physical DNA from the distribution plates, and we generated 17 new biobricks, which then we produced working containment modules. We screened two previous iGEM projects for existing safety modules. We built our new containment modules, and from this we created a biosafety page in the registry, so you can easily find the biosafety modules that suits for your project. And we interview experts, we engage public, we collaborate with iGEM teams, and from our human practice project, we are giving proposal and opportunity for collaboration, which we hope can be useful for all of you. So thank you to our sponsors, and thank you all for your attention.
Thanks very much. I see some questions already.
So thanks very much. A very interesting project and a very clear presentation. I'm curious because you seem to have started your entire project from the premise that it's a bad thing if these genetically modified bacteria or genetically modified genes escape into the environment. And if we go back to the first presentation of the session, and imagine, say, a catabolic operon that could degrade these endocrine receptors and these kind of modified estrogen compounds. Basically, they're xenobiotics, so they shouldn't be naturally occurring in the environment. But say catabolic operons that could degrade them were to actually just get out and be prevalent, say, in our wastewater treatment plants, just on accident. Well, then we might see natural, degradation of these man-made compounds, and in fact, our wastewater might be maybe safer as a result. So can you just comment on kind of, how dangerous do you think it would be if some of these, bioremediation systems were to actually just escape and just be in the environment, particularly the ones that are degrading xenobiotics?
Well, I think this is some kind of best case scenario if we provide natural population with bioremediation tool, but this is kind of madness to think that nature will act exactly what we want it to act. So we need to be really careful about the way we introduce genes in the environment, as the consequences are completely unpredictable.
So here we think, the point where we don't know what will happen next is the point where there's host-role gene transfer. So that's the limit we put for safety. Then we think, as we said, case-by-case project. If people want to to decrease this barrier for safety and can prove that in their case for their project it's not going to be something bad, we think they have the burden of proof. We set the bar for safety quite high. And then we think, most importantly, that it's the public's decision to take, so they should be informed, they should have, risk and all this kind of stuff should be well quantified, they should have good information, and then it's for the public to decide if they want or they don't want this project, and again on a case-by-case basis.
Okay, but so to follow up, say we have a really high bar for release of engineered organisms into the environment. So that means that it's going to be hard for some, say, iGEM teams to ever get their projects deployed. It might be hard for some companies to ever get their products out in the market. So that means that we might not be actually cleaning up some of these pollutants in the environment that we would maybe otherwise be able to do so if the bar was lower. So, I mean, have you just considered, so I agree, there's the danger of release of engineered organisms, but there's also the danger of not releasing organisms and therefore not accruing some of the benefits of release of engineered organisms.
That's why we're firstly interested in this, because there are so many interesting projects that it's like there are these huge benefits. But we can provide numbers and we can provide suggestions, but where the bar is set is not up to us also. So if people see that the bar is too high, the public wants it lowered, that we can just provide numbers and let the public decide, and the scientific community.
So I've been involved in a number of policy discussions that occur at the relatively qualitative and abstract level, and where people often say, well, it would be nice if we had some numbers on these. And for example, your estimates of bounds and prediction of how the different components of your systems might fail and they have potential aggregate failure rate, it's nice to see that. And that's also very well detailed on Wiki. So that which I've only partly read through that section. But it's got the detail that I'm looking for. When you give your estimate of, say, 10 to the minus 11th order of magnitude failure rate of the system per generation, am I right in understanding that that's from the perspective of per cell, each individual cell's chance of that occurring? Or,
yes.
And if that's the case, does that lead to a recommendation of how many cells one might want to be working with at a time. So, for example, 10 to the 11th cells, which is not hard to achieve in a reactor, if the containment system was absolutely necessary, is that what you would, I guess what I'm really getting is how would you turn these into recommendations for people?
So the problem with this number is like there are many things or there are many events that are still hard to predict. Like for example, what are like the population level suicide is, I mean, we put just one as a worst case scenario, but actually we have to take into account this, like what is the density of the population and how effective is population self-suicide, and yeah, and that, you want to answer?
This is a worst case estimate.
Yeah, so it's a worst case estimate.
I had a question about your colicin population killing. You mentioned that it was robust to a certain level of cell in the population losing the ability to make the colicin. Do you know what fraction of the cells can lose the colicin making ability and still get robust population killing?
So like how many, like what are the probability of the colicin lose the?
I think I understand your question, just that, so we could have a case where a cell doesn't like produce the toxin but is still killed, and so we don't have numerical numbers for that, but what we wanted to try is using the colicin extract, use different concentrations, and see at what point we'll still have cell death even in the presence of immunity. So we basically bombard the cells with colicin and how much it takes to do that. That was an experiment we would have liked to do.
I also had a question about your containment system. You had great improvement for the amount of escaping cells, but you were still seeing thousands of cells escaping per bead or per sample. And how do you feel about that result?
Well, of course, we have to decide what is an acceptable risk and incorporate that into the assessment of risk on top of the other levels of containment. So, since every level of containment is only an incremental increase, we try to use the capsules as just one of those incremental improvements.
I think we have one question from the audience.
Regarding your colicin safety procedure, I was wondering if you happen to know of any other environment, bacteria or organisms or whatever, also actively internalize that colicin, and that may be an undesirable effect of your biosafety.
So the question is that you are wondering if our colicin is very specific to E. coli? Or,
Yeah.
Okay. So it is specific to E. coli. It's also imported by very similar species, but normally bacteria in the environment are not the same as the one in your gut. So we do not expect any import and any toxicity to naturally occurring soil bacteria. Could happen.
I'd suggest that further audience questions you might want to discuss with the team afterwards. I'd like to end the session by thanking all of the speakers again.